Hello, everyone. I hope you had a lovely weekend. Welcome back to the Helios Speaker Series, the fifth speaker series so far. For those who are not familiar with the Helios Project yet, the Helios Project is an EU-funded project over 5 years, and our goal is really to standardize the care for hemoglobinopathies by bringing together experts in the field, providing networking and training possibilities, and then disseminating the knowledge created. If you want to find out more about Helios, please visit our website, heliosaction.eu. And our website, you also find more information about upcoming events like the speaker series. For our Helios speaker series, the next speaker will be Dr. Immaculata Andolfo, who will talk about her Piezo 1 work. If you want to join her webinar, please register for this webinar individually as well, as you have to register for each webinar separately. On the website, you will then also find the recordings of past presentations, such as from Agnieszka Schipper, who spoke about her AI approach for the early diagnosis of hemoglobinopathies, and Dr. Kristen Howell, who spoke about challenges in the transition from pediatric to adult care. And today I'm really excited to welcome Dr. David Williams. Dr. Williams is an internationally recognized researcher focused on blood stem cell biology, leukemia, and gene therapy for genetic blood disorders. He's a professor of pediatrics at Harvard Medical School and chief of hematology oncology at Boston Children's Hospital. Dr. Williams' lab has been funded by the NIH since 1986, reflecting his significant contributions to medical research. Throughout his career, he has mentored over 40 postdocs, many of whom have established their own labs or joined biotech or pharmaceutical companies. Today, I'm excited to welcome Dr. Williams to talk about his recent work on the UM171 molecule, reducing the stress in HSCs and increasing HSC fitness. Dr. Williams, thank you so much for joining today. It's really exciting to have you with us today, and we very much look forward to your presentation. Thank you. Over to you.
Thank you for inviting me, and good afternoon from snowy Boston. I'm happy to be here. I've kept this talk to about a half hour so that there's time for questions at the end. And I will talk about what we're doing to try to address one of the many issues that still are impediments to the use of gene therapy, ex vivo gene therapy in sickle cell disease. So here are my disclosures. There's none that are particularly relevant for today's talk, except that I will point out that I'll be talking about a clinical trial we're doing in which the GMP vector for the trial was provided to us. It's our vector, but the Bluebird Bio actually produced the GMP vector for the trial. So I'll talk a little bit very quickly about sickle cell disease, gene therapy. I'll talk very quickly about the development of a novel approach to targeting a transcription factor called BCL11A using a lentivirus vector containing a schmear. For that, I'll talk a moment about the whole concept of fetal hemoglobin and sickle cell disease. I'll talk about the pilot first-in-human studies that we performed using this schmear vector. I'll touch on the ongoing pivotal phase 2 multi-site trial, and then spend more time on ongoing work, particularly work that we're doing to try to reduce stress on hematopoietic cells during ex vivo manipulation and manufacturing. And this is relevant not just to lentiviral gene therapy trials, but also to editing trials in which the cells are manipulated ex vivo. So as you know, autologous gene therapy approaches for beta hemoglobinopathies have really progressed dramatically over the last several decades. Bluebird gene addition called Lyfgenia has been approved by the FDA for sickle cell disease. You probably know that the CRISPR gene editing approach using Casgevy has been also approved. We are a center for base editing by the sponsor Beam Therapeutics, and we have our own gene editing trial in progress led by Dan Bauer. And of course, in development but not yet deployed is actual correction of the sickle mutation. And what I'll talk about today is this approach called the Schmier knockdown of BCL11A. So many of these approaches focus on fetal hemoglobin, and one question you could ask is, why is all this focus on fetal hemoglobin in sickle cell disease? It's been known for a long time that individuals that have high levels of fetal hemoglobin have somewhat attenuated disease phenotype. And actually, the first observation regarding this was done by a pediatrician named Janet Watson in New York. You can see the date, 1948. She noticed that in babies that were born of mothers with sickle cell disease didn't really display signs of sickle cell disease. And in fact, their blood films showed many less irreversibly sickled cells than their mother. And so she hypothesized that this must have something to do with the fact that they express fetal hemoglobin rather than adult hemoglobin. Of course, that's correct. Here is a chart showing you, for those of you that aren't following or aren't hematologists, the expression of hemoglobins during development and in adulthood. And prenatally, you express, of course, alpha globins, but you express, instead of adult beta globin, you express gamma globin. And so alpha and gamma make up the fetal hemoglobin molecule. After birth, you switch off gamma globin expression and you turn on beta globin expression. And of course, for individuals with sickle cell disease, that means you go from having no sickle cell and no symptoms to turning on the beta locus, which is contains the sickle mutation. And when I see babies in the clinic in the first 6 months of age, they rarely have any difficulties. But after that time point, up to around age 12 months, then they begin to develop sickle symptoms. Now, this was also shown by multiple investigators over time. You can see here, if we look at just pain, another symptoms of vaso-occlusion called acute chest syndrome, and death, that the higher you have, your higher your fetal hemoglobin, which in most individuals is less than 1% as adults, the less of these complications. So the less pain episodes you have, the less acute chest syndromes you experience. And in fact, just a difference of fetal hemoglobin levels of less than 9% versus above 9% in individuals with sickle cell disease can add a full decade of life on life expectancy. So it was known for many years that the level of hemoglobin F was important with respect to the disease. And this was also true in populations throughout the world. For instance, populations in Saudi Arabia and some populations in South Asia, which have high levels of fetal hemoglobin, characteristically have a little bit less symptoms of sickle cell disease. But the breakthrough in the field came from GWAS studies shown here, a Manhattan plot showing that in addition to sort of expected loci that affected fetal hemoglobin, there's this loci called BCL11A. It's B-cell leukemia 11A. It's a transcription factor that's required for B-cell development. But before this study, there was no indication that fetal hemoglobin would be regulated in any way by this B-cell transcription factor. And Stuart Orkin's lab went on to show actually that you could, in the mouse system, reverse this switch. So after birth, shown here, the regulatory elements that control expression of the beta globin locus in utero, these locus control regions interact with the gamma globin gene here. After birth, you can see that they now interact with the adult beta globin locus, and of course in sickle cell patients, that's beta sickle. So the idea would be the therapeutic goal would be to reverse this, and Stu showed in his studies that if you looked at a mouse knockout of BCL11A, you could in fact almost completely reverse the sickle cell phenotype in the mouse. So our approach and goal was to rapidly translate that basic science discovery into treatment for sickle cell, doing translational studies and then clinical studies using the available viral vector platforms, which of course really at this point was lentivirus vectors. So we wanted to knock down BCL11A using RNAi to induce gamma globin expression and silence the mutated sickle expression. And so we're thinking about this as essentially a physiologic binary switch where we can turn off BCL11A and increase fetal hemoglobin expression. And the advantage of that is the fetal hemoglobin actually has anti-sickling characteristics. So it inhibits the polymerization of deoxygenated sickle that's in the cell. And at the same time, because it's a binary switch, you downregulate the beta sickle expression in the cell. And so using that physiologic switch, the alpha and beta globin genes remains balanced. This is a little bit of a subtle point, but an important one. This is in contrast to gene addition, where you would express more normal hemoglobin, but not necessarily reduce the amount of sickle hemoglobin concurrently. So to do this, we developed a lentiviral vector called a Schmier vector. We embedded the shRNA targeting, the optimal shRNA targeting BCL11A in a microRNA architecture so that the cell actually processed this instead of as an shRNA, it processed this as an endogenous microRNA hairpin, shown sort of schematically here. And the advantage of that, of course, is it mimics a microRNA rather than an shRNA, and it leads to more physiologic and lower expressions. And that also then leads to less non-specific toxicities. You may know that shRNAs, when expressed at high levels, have toxicities in the cell. But probably more importantly for us, it actually allows us to regulate expression only in erythroid cells. And that avoids the toxicities that you can envision for a transcription factor that's required for B cell development in B cells. But also we discovered in these basic science studies that BCL11A is required for appropriate stem cell engraftment. And so this allows us then to develop this erythroid-specific cis-regulatory element containing enhancers called HS2 and HS3, the minimal beta promoter, and then expressing this smear technology. So that led to, and I'll just point out that there are multiple publications if you're interested in the technology shown here. So that led to a first-in-human feasibility study that was a single-site study, originally 7 patients, and then we increased to 10. And this study is completed at this point. And the inclusion criteria included a lack of a matched sibling donor because that's the standard of care for sickle cell patients undergoing an allotransplant, a severe phenotype shown here, a failure for hydroxyurea. And originally we included patients with a history of stroke, but we subsequently modified that to exclude those patients. And the study design is pretty straightforward for a lentiviral ex vivo trial. We mobilize cells in the peripheral blood with an agent called plerixafor. Those cells were CD34 selected in a GMP facility, transduced and cryopreserved. And then once the release criteria were arranged, the patient was admitted to the hospital for ablative conditioning with single agent busulfan. And then the cells were infused and the patients had follow-up. So this original study has been published in January 2021, shown here. We call this post-transcriptional genetic silencing. and the study was quite successful. This study led us to have some important correlative biology also that I'll just point out for those of you interested. One is that we did a study in collaboration with John Higgins at Mass General Hospital, in which we actually analyzed at a single cell level the behavior of deoxygenated sickle hemoglobin polymerizing in lower oxygen concentrations. And we compared patients in the gene therapy study with those getting standard hydroxyurea treatment that's shown here. And then we also looked at single cell analysis on the effect of cell manufacturing, vector transduction, and engraftment on mutation burden in sickle cell disease patients. And this is in collaboration with Mike Chapman and individuals at York University in the UK. This led to a lot of interesting studies in which we continue to ask the question, are patients with sickle cell have more baseline mutations due to their disease, or do they have a higher risk of developing MDS or leukemia during gene therapy? I'm going to talk a little bit more about that. But this has led then to continued studies funded by the Gates Foundation, in which we're looking at the genomic landscape of sickle cell disease compared to ethnic control populations in the UK, in Boston, and in Tanzania and Uganda. And also the NHLBI is funding a large-scale genomics project with Coleman Lindsley at Brigham and Women's Hospital that we're participating in with our study also. So these studies are ongoing and haven't yet reached maturity. All these studies in the phase 1 trial led to a phase 2 pivotal trial called GRASP. It's Gene Therapy to Reduce All Sickle Pain. This is R01 funded and Cure Sickle funded from the NHLBI and the California Institute of Regenerative Medicine. And I'm co-chair with Mark Walters at UCSF. And the primary endpoint of this is to determine if a single infusion of these autologous transduced cells with the Schmier vector can lead to complete absence of severe VOEs in the period of 6 months to 24 months after gene therapy. And if this trial hits its primary endpoint, then by our negotiation with the FDA, we would be able to file a BLA for the use of this in the future as a drug treatment. Now, this is a multi-site trial. I won't go through this, but here are the sites. And currently, the trial was activated in January 2023. There was a year hold by the funder, NIH, because of the leukemias that occurred in the Bluebird trial, although that really is a different vector and it really is different than our trial. But in spite of that, we've enrolled 31 patients, 25 have passed screening, and then that's our accrual target, so we're done recruiting. In this trial, we've been able to basically validate a second lot of vector that was introduced that's made in a commercially compatible facility. We've had 22 products manufactured, 17 patients have been infused. There's been no safety signals to date, and so we're quite pleased with the study, although it's ongoing, of course. And we had additional genomic surveillance. I mentioned this earlier. So both pre and post infusion, we're monitoring genes associated with myeloid malignancy and MDS, and they're listed here. And there's an expert independent genomics panel that adjudicates these studies. And to date, of the 25 patients that we've enrolled, we've had no patients screened that had a pathogenic mutation identified. So moving forward, we're working on several different fronts. I'll talk a little bit about reducing genotoxicity. We've developed newer vectors and enhance even further F induction and concomitantly reduce concentration of sickle hemoglobin. This is a collaboration with Don Cohn using our basic Schmier vector and his modified regulatory elements and modified beta globin cDNA. I'm sure many of you know Don or know of his work. And then we're developing in vivo delivery in a large project that's been funded by the Gates Foundation, and that's a collaboration with Christian Brendel, who's one of our junior faculty who came originally from Germany to our program. So why worry about genotoxicity? Just to remind you that in 2021, Bluebird announced a temporary suspension of their gene addition trial, which was in phase 3, because 2 patients developed AML. And these were unexpected safety events, but they were not apparently related to vector insertion. One of the patients had no vector inserted in their blast cells at all. The other patient had insertion in their blast cells, but a lot of work by Bluebird definitively shown, or as definitively as they could, that that insertion was not related to the oncogenesis at all. So the question really became, does in vitro culture for transduction and editing select cells that have pre-existing mutations favoring proliferation and survival? And does lentiviral transduction or gene editing itself cause genotoxicity? And we explore these sort of thoughts in this paper I mentioned already, published in Nature Medicine last year. And so the question became, as we were doing genomic studies, could we further reduce stress during manufacturing? Because, of course, when you put hematopoietic stem cells in culture, even for hours, and then ask them to engraft into a recipient, all those manipulations can potentially increase the stress of the stem cell itself. So we began relatively quickly to collaborate with Guy Samia, who many of you may know, who had published in the past pyrimidinol derivative that he showed had an effect on expansion of long-term hematopoietic stem cells. So here's the references for your knowledge, but here's a figure from one of those papers in which he looks at primitive stem cells by immunophenotype and increasing concentrations of UM171. And you can see that in vitro, there's a definitive increase in the number of these immunophenotypically defined stem cells. And there's a lot of data subsequently using cord blood in clinical trials to show that this, in fact, does enhance the engraftment of these primitive cells. So our question was, could this be applied to sickle cell disease? And so this work has just recently been published by Boya Liu, who's a postdoc in my lab. And for these studies, she would compare cultures with and without UM71 in a standard lentiviral transduction protocol. And the engraftment was analyzed in immunodeficient mice, MB-SGW mice. And so if you look at these studies, if you look at engraftment 16 weeks after infusion by the number of human cells, or if you looked at the engraftment by the number of gene marked cells, using our standard transduction protocol with either a control vector with GFP or GFP with UM71, you see a significant increase in the engraftment of the manipulated and transduced cells. So we then looked at a derivative of our research derivative of our vector that's in clinical trials. So here again is the B7-A schmear. It's in a microRNA called 223. It has cis regulatory elements we mentioned before, HS2 and HS3, and a minimal beta promoter. And so in this case, we looked at a direct comparison, comparative repopulation with vectors that have either a Venus tag or a BFP tag, and then we can either culture with or without UM71, and then these cells are co-injected into the same mouse. And you can see here that if you look at the percent of all gene-marked cells in the peripheral blood, you can see over time up to 16 weeks, but even from the beginning, there's an advantage of UM171 manufactured cells. And when you look at the ratio of the marked cells with or without, this is a log scale, you see a very significant increase, interestingly, over time, suggesting that the effect that we're having is actually quite dramatic on the more primitive cell that reads out longer term in the mouse. So then we ask the question, what's the basis, the mechanism of this increased engraftment? And so we looked at three different parameters shown here. One is actual direct DNA damage using gamma H2AX, a common assay, flow assay for DNA damage. We asked the question of, was there an effect on the development of reactive oxygen species, which can affect DNA damage? And then we asked the question of apoptosis using annexin. And you can see in all three of these assays, there's a significant reduction in apparent damage to the stem cell population when we do the transduction in the presence of UM171. We also asked the question, what about polyclonality or clonality in the recipient animals? And these are sort of standard stacked bar graphs showing in each individual case column a different mouse, and the barcodes show different stem cell contributions to the mouse's peripheral blood. And you can visually see the difference with UM171. And then this is quantitated by Shannon diversity scale here, so that you can see there's a significant increase in stem cell diversity, essentially meaning that there are more stem cells repopulating the animal, and there's therefore more clonal diversity in the recipient when UM171 is used for into production ex vivo. So the conclusions of that work are that the culture of sickle, these are CD34 cells derived from sickle cell patients during ex vivo genetic manipulation, significantly expands the stem cells, enhances transduction, reduces DNA damage and reactive oxygen species generation, therefore increases the engraftment of those cells and shows increased clonal diversity and clonal balance in those recipient animals. So these data suggest that UM171 should be tested in human gene therapy trials in sickle cell, and it may be applicable in other ex vivo gene therapy trials. So while we were particularly focused on sickle cell disease, it's likely that any stem cell population that's being ex vivo manipulated, either by gene transfer using lentivirus or any sort of gene editing, would likewise sustain DNA damage. I'll just move quickly before I end up with a couple comments. I mentioned already that we have now modified our vector to further induce F induction and reduce concentration of sickle cell in a collaboration with Don Cohen. I won't go into these data, they're both published here. So the first was to show that we could tandemly arraign multiple smears targeting different genes. And in this case, we did both B7A and another transcription factor called ZNF410. And by doing those double smears, we could further induce fetal hemoglobin induction by about, on average, 10% in cells, which is clinically significant, as shown in this paper. And then with Don's work, we actually developed a bifunctional lentivirus vector that contained these double smears and added expression of his sickle-resistant beta globin mutant cDNA. And essentially, the bottom line of this paper is that using that approach, we can reduce the hemolysis rate in mice with sickle cell, model mice to basically control levels. So it's as if there's no sickle functional anymore at all in the cells. So this is quite exciting, and we're hoping to develop that further. And then I'll just touch on one more topic, which is developing in vivo delivery. And to that work with Christian Brendel, we basically developed baboon EV pseudotype lentiviral vector. And to produce this, we generated producer cell lines that were knocked out in the receptor for this virus, which are two genes, ASCT1 and ASCT2. And you can see we were able to generate reasonable titers from this. We could get very significant transduction of CD34 cells that were quiescent. And interestingly, this pseudotype is resistant to serum inactivation, which is an important constraint when you're talking about in vivo gene therapy. And we've been able now to show that this works in the mouse. So we engraft human cells into the xenograft model. We can mobilize these cells using a combination of AMD 3100 and GRO beta, and then directly inject intravenously the lentiviral vector. And then we've developed into this vector a cDNA for MGMT. This is methylguanine methyltransferase that encodes chemoresistance to BCNU. And actually, interestingly, this was a approach that I developed as a very new faculty member in 1986 when I started my own laboratory at Boston Children's. So it's been around for a while, further really developed by Hans-Peter Keim in Seattle. And using this approach, interestingly, we can get, as you can see here, up to over 60, almost 70% of bone marrow cells with a single infusion of virus intravenously in these animals, in human cells. And this includes, if we isolate at week 12 CD34 human cells from the bone marrow, 40% transduction of those cells. And this BCNU selection allows us to get about a 60-fold enrichment of transduced cells. And now we've gone on to incorporate our double smears into this vector. And you can see, if you just look at this slide on the right, that you can get over 50%, almost 60% fetal hemoglobin in these cells from animals that are transduced in vivo. So we're quite excited. This is all unpublished. We're quite excited about this work and are continuing to develop this with Gates Foundation support. So the overall summary is that introduction of the smear technology appears to be both safe and effective. There's no evidence of any genotoxicity. I didn't go into this data, but we now have follow-up over 5 years in multiple patients with no clonal dominance at all. The cell manufacturing has been very consistent, and unlike gene editing, we can generally get enough cells for manufacturing with one mobilization cycle rather than multiple. The pivotal trial is ongoing and targets accrual has been obtained. Excuse me, and we need to have additional studies to define genomics on this group of patients, and those are underway. We've now incorporated multiple process improvements that may increase efficacy further, and these may be applicable to other ex vivo approaches. And I showed you a little bit of preliminary evidence for the potential for development of in vivo approach using modified viral vectors. So I'll stop. I know I wanted to leave time for questions. This is, of course, a major undertaking to go from the bench to the bedside. We've had enormous amount of work, including patients from the sickle cell community who've volunteered for these studies. At Boston Children's, our PI is Erica Ezrich, but we have major help from the other sickle cell docs and from our translational lab led by Miriam Marmont. and our GMP cell processing lab at Dana-Farber Cancer Institute. We've collaborated, as I mentioned, with John Higgins at MGH, with Pablo Bartolucci in Paris, and had great collaborations with Bluebird Bio. And of course, the funding I've mentioned already. The phase 2 study is run through BMT-CTN. It's funded by, as I mentioned, NHLBI. The protocol officer is Mary Epen. The coordinating center is Boston Children's, and the data coordinating center is the Medical College of Wisconsin. And we're manufacturing both at TFCI and at UCLA. And then my own laboratory is relatively small, but Chad Harris is my lab manager for over 26 years. And I mentioned already Boya Liu, who's a postdoc who did much of the work, our collaboration with XL Therapy, which is the company started by Guy Sauvignon. And mentioned already the collaboration with Christian Brendel on the in vivo approach. The gene therapy program at Boston Children's is an amazing program. You can see individuals here led by Dan Bauer and Christy Duncan. We have both a clinical research team, and we have a number of clinical investigators, and we collaborate with a number of biotech companies. As I mentioned already, we use the GMP lab at DFCI. Okay, so I'll stop there. Excuse me. And
Thank you, Dr. Williams. Really fascinating. Yep. Wow, like it's really promising, obviously, to hear about your work. And we now open the floor to questions. If you either want to put your question in the chat or if you want to raise your hand. I'm really curious, like, to kind of start off with, like, have you done any work in thalassemia as well? Because obviously these patients have a lot of hope when it comes to gene therapies as well.
Yeah, we've been focused on sickle cell disease. But I mentioned a gene editing trial that we're opening with Dan Bauer. That will include thalassemia patients. And several of the commercial products, of course, use the same vector for both thalassemia patients and sickle cell patients. And theoretically, the vector I've described, the Schmier vector, should work quite well in thalassemia. We've just really been trying to focus on the sickle disease group.
That made sense, of course. We got a question from Marie Sebastian. Marie, do you want to ask the question? I think you have to unmute yourself.
Do you hear me?
Yes, perfect. Wonderful.
Thank you for the talk. Marvelous. One question about the clonal diversity of the stem cells. First, which marker do you use to distinguish clones of stem cells? And second, why is it good to have clonal diversity in this specific field? One may think that a few clones will reduce the risk to have a genotoxic event thereafter.
Yeah, that's an excellent question. So for viral vector trials that integrate, and so because these are hematopoietic stem cells that proliferate a lot, traditionally integrating vectors have been used. The integration site basically marks the stem cell at a clonal level. And we and everybody else that does these trials carefully follows the integration profile. Excuse me. And that can tell us the clonality. For the trials that we do in the research lab, we can also add a barcode to the vector and easily then use that barcode to follow the populations. But traditionally in clinical trials, we use the integration site analysis. And what we find is that, or what the hypothesis is that if you have fewer clones in grafting and requiring those few clones to sustain hematopoiesis over a long period of time, then you put more stress on those clones, which could then lead to selection for clones that have integrations in genes that somehow affect the fitness of the stem cell. And usually that means it affects their proliferation. So the theory is that you're better off with a highly polyclonal population of cells. And just it doesn't prove that theory, but in the old gamma retroviral vector trials, and so I started out my career using gamma retroviruses. If you remember, there were leukemias in the SCD trial in Paris and London, and then there were subsequently leukemias in the Wiscott trials in Germany and the CGD trials in Germany and Austria, or no, not Austria. I forget the other country that trial was done in. Those were associated with integrations into oncogenes, and when you followed integration patterns and polyclonality over time, what we saw was that the clones that had those integrations outgrew everything else. So that with time, the engraftment became more and more oligoclonal until the point where it was almost monoclonal. And we recently published a paper in New England Journal on the adrenoleukodystrophy trial, in which it's a lentivirus, but it has an LTR in the vector. And we also saw leukemias in those trials. And in those cases, we also saw the evolution of oligoclonality prior to the development of leukemias and MDS. So that's kind of a long-winded answer to your question, but that's the reasoning.
Yes, and the second question was regarding engraftment. If I understood well, your small molecule is associated with better engraftment of stem cells.
Yes.
Do you think this could open the way to reduce the conditioning regimen, which might be the main factor of clonal evolution of stem cells, especially in patients in whom pre-existing clonal mutation are detected?
Yeah. I think that the effect is not so profound to allow one to approach engraftment without any conditioning at all.
Mm-hmm.
I think that's a really important point, because the major impediment now to wide development of gene therapy in many diseases is the fact that the ex vivo approach requires basically an ICU type admission for 30 to 40 days if you're lucky, and immunosuppression drugs sometimes, and not always, but certainly alkylating agents to get rid of the endogenous hematopoiesis. So I think the field is waiting for the further development of immunologic conditioning, which would not require chemotherapy. I don't think that the use of the UM171 is enhances engraftment enough to eliminate conditioning, basically.
Maybe just reduce, which might be a good option, as we are doing in the allo transplant setting, where we can reduce to 3 or 2 days of busulfan rather than 4.
Yeah, it's possible. The caution there is that in the NIH has reported in sickle cell disease leukemia development in allotransplanted patients with sickle cell disease, and it's uniformly in patients who've had reduced conditioning. And the theory is the reduced conditioning allows damage to stem cells that are not then eliminated from the bone marrow, and those damaged stem cells then contribute to the evolution of MDS and leukemia. it's a theory, but so I think there's some worry about non-complete conditioning in the setting where you have some potential already endogenous damage. For instance, in Fanconi anemia, which is a disease I take care of clinically, those patients have likely already at birth damaged stem cell populations. And if you didn't get rid of their endogenous hematopoiesis, and you did gene therapy, for instance, or you didn't get rid of their endogenous hematopoiesis and you did an allograft, they could have residual damaged stem cells that then, because of their disease phenotype, would potentially have a propensity to develop MDS and leukemia. That's a pre-cancer syndrome. So their stem cells, like every other cell in the body, has problems in repairing DNA damage.
Okay, thank you. By the way, I'm working with Pablo Bartolucci in Paris. He could not be there, but he asked me to send you his warm.
Oh yeah, tell him hi for me. He's a great guy. Thanks.
Wonderful. Could I ask on that note, because you obviously just mentioned the choice of vector and the risk of developing MDS and leukemia. It sounds like it's not so much about the choice of vector, but it could be down to the residual damage that already exists because of the gene mutations that they have because of their condition. Is that correct, or what are your thoughts on that?
I think that's still a question, and so I think one question that the field is very interested in is, because of the inflammatory milieu of the bone marrow in patients with sickle cell disease, is there some already DNA damage that would potentially increase with time as you become an adult, that is part of the slight increase in risk in leukemia that's been seen in a couple natural history studies in that disease. But I think we don't know yet, basically, and that's why we need some more ongoing genomic information.
And you also mentioned at the end of your presentation, I'm sorry, I don't want to dominate. If anyone has any other questions, please put them in the chat or obviously raise your hands. But I'm curious, obviously, because from a patient's point of view, they'd be obviously curious to hear about any studies you might be with in terms of obviously using UM171 in clinical trials. Have you heard of any transition into human studies?
Well, we're working with Excel there and a couple other groups to try to effect an IND that would allow us to incorporate that into a small pilot feasibility study. It's already been used in ex vivo expansion of cord blood, but specifically in gene therapy hasn't been used yet. We would really like to do it. And so we're working to try to accomplish that. It's not ready for prime time yet, but we've had a pre-IND meeting with the FDA, and we're working on the studies that are required to get us over the finish line on that.
That's very exciting. So we definitely look out for that work then. Does anybody else have any questions? If you want to put them either in the chat or raise your hand. We can give it another minute or so if you want to think about it. Obviously, if you got any questions following this live presentation, if people are obviously watching the recording, how can they best get in touch with you, Dr. Williams, if they have any follow-up questions?
You can just use my email address. It's I don't know if you have it posted somewhere,
Yes.
but okay. Yeah, they can just email me. It's
Wonderful,
totally
very
fine.
good then. Okay, very good. So unless there are any other questions, so the recording will be made available on our YouTube channel, which I'm sure obviously a lot of people will watch, and then they might get in touch with you, Dr. Williams, if they have any follow-up questions. But thank you so much for your presentation today.
Thanks for inviting
really
me.
fascinating.
Yeah, thanks for inviting me.
And all the very best for your ongoing work. We're getting a few more people saying in the chat, excellent work and great results. So thank you so much.
You're welcome. Thanks.
And all the very best to you. Thank
Okay,
you.
bye.
And thank you everyone for joining. Take care. Bye-bye.